Hello, good afternoon, and welcome to the second topic of our My Wine seminar. Today we have the history, the origin, and the future of Grüner Veltliner. Thank you for all your interest and that you are participating. And José, welcome, and thank you for preparing all the slides. We will again offer you the slides on our website from tomorrow on, and you can download the PDFs. And we will also offer you a video replay function for this seminar. So if you would like to hear something again, or those people who could not participate, they will have the option to download it from our website. When we've set up the Zoom meeting, all your microphones should be on mute. And as we have a bigger number of participants, we ask you to keep it on mute till the end of the presentation. We have afterwards a Q&A session, which takes as long as you want. José will take his time, but we will not interrupt him during the presentation, because otherwise there might be on some topics too many questions. And we try to stick to this 45 minutes program. So the first 45 minutes are the presentation and then the Q&A session. Just as the beginning, because some will drop off afterwards immediately before I can make my closing comments. We are planning to extend these sessions next year. With José, he has suggested some other ideas. One would be the Cabernet family, including Malbec and Carmenere. Another topic could be Syrah plus grape domestication and the origin of viticulture. And a third topic we have discussed would be Blaufränkisch and Eastern European grape varieties. We plan this next year again via a Zoom meeting, but for those who are in Vienna or close to Vienna, we also plan physical meetings if this is then allowed, and serve wines alongside the topic of the seminar. But now let's come back to Grüner Veltliner. We in Austria say Grüner Veltliner, but in the invitation we always write Grüner Veltliner because we have so many international people there. And José, I would like to hand over the stage to you.
Thank you very much for this nice introduction. Hello everyone, I'm very happy to be with you this afternoon. It's always a pleasure to see so many people taking the time to speak about our common passion, which is wine and today grapes. Thank you again, Bernd and maywines.com for inviting me. It's a real pleasure to share my knowledge with you. And I'm looking forward for the future presentation. And today I'm sharing my screen with you. I need to take the correct one, Grüner Veltliner. We will speak about Grüner Veltliner, so I'm gonna go full screen. I think everybody's seeing the screen right now. So, as a teaser, I wanted to tell you that we're gonna speak about sex. It's always easy to catch the attention of people with the word sex anywhere. It's working, I'm telling you. So yes, we will speak about grape sex because I had a few questions last week about that, and it's interesting to set the things up. Then Grüner Veltliner will look at the history and origin and also the future. Speaking about the future, I will need to address the question of GMOs. I know that many of my colleagues do not want to talk about that. It's a hot topic. I want to address this question in a neutral way. I mean, I'm in Switzerland, we are neutral. So I just give the information that we have, and I will end with the special technique called gene editing. Again, I'm not going to go into much details. You don't need to be scientist or geneticist to understand. It's just to have a general idea of the topic. Let's talk about grape sex. If you have ever seen grape in flower, you have the cluster with all these, and I will try to point at this here. Yes, all these small buds. Actually, these buds are flower buds, and it's a baby flower. It's the flower that will open. And if you look closely at this, then I will show you a picture. You see that as a regular flower, the grape flower, it's very, very small. It has petals, but those petals are bound together. They are stuck together, and we call it calyptra or flower cap. So it's a cap over the flower because the petals are stuck together. And instead of opening like a rose or like a tulip, it does not open up, it opens from the bottom. So you see that here on top, it cannot open, so it detaches from the bottom here to open the flower. Once this cap is removed, that means the petals are removed, you see the structure of the flower. This big stuff here is the female part. Here you have the stigma, is the female part that will receive the pollen. And this is the ovary, inside which you have the embryo that will develop into a seed. So stigma is here. The 5 branches here are the stamens. The stamens with this bag containing the pollen. So the pollen is here in each bag, and you have the stigma that receive the pollen. What's happening here? You have the pollen that is ready that goes on the stigma. Then you have a tube going down to fertilize the embryo, to fertilize the egg and develop into grape pips. And all this stuff will become a berry. So this is how sexuality is working in grapes. You can see it with real pictures. Here we have a picture A when the cap is dried and is going out by itself. And then here it's out. Here you have the free flower, the female part in green, and the male part with the pollen bags that will give the pollen. So that's, if you look closely at the grape flower with a magnifier, that is what you will see. Now, what's happening? It's back to the questions we had last week. I made this simplified slide to explain what are natives versus crossings versus hybrids. Natives on the left-hand side are spontaneous crossings that happen naturally in the vineyard. So it doesn't need human intervention. For natives, you have 2 possibilities. On top, you have selfing. That means when the flower is mature, it could even be before the cap is off. If the stamens here are ripe, the pollen will be produced and fall onto its own female part, its own pistil. This is selfing. So the grape flower will pollinize itself. And we have no actual figure, but we estimate that this is the majority of cases, which explains why we have harvest everywhere, every year. But it also happens that the pollen is transported from one flower, from one flower here to the other, either by insects. Here you have a bee that is on nectar. It's not a grape flower, it's just to have a funny sketch, either by insects or by wind. We have observed bees and dipters on grape flowers, but the production of nectar, which is the juice that attracts the insect, is not so big in grape flowers. It's much bigger in other botanical species. So again, we don't have any exact figure, but we estimate that both are playing a role, insects and wind, to bring the pollen from one flower to the other flower. So the pollen from here onto the pistil, so the female part of the other flower. This is cross-pollination. When this cross-pollination happens today, most of the time in the same vineyard, you have the same variety. So even if I have two different plants, here I have one plant of Pinot, for example, this other plant will most likely be Pinot. So we would still have Pinot fertilizing Pinot, and the result is the same as here, genetically speaking. What is interesting is when you have two different plants, two different varieties next to each other. And here you can have, let's say, I go back down. Here you can have Pinot, and here you have Gouais Blanc. We spoke about that last week, which created Chardonnay. So if you have two distinct varieties pollinating one the other, it's much more interesting, and the result is much more interesting. And for example, it has been Chardonnay, Gamay, and so on. So this is what happens without human hand. On the other column, you have controlled grape breeding. That is what is carried out in research stations in experimental vineyards. I separated two categories. The first one is crossings. Crossings, for me, and what we have published in Wine Grapes and the general understanding, crossings is one variety from Vitis vinifera, the European grape, crossed with another variety of European grape, Vitis vinifera. So we remain within the same species. So you can take here, you can take one variety, pollinate the other, you have a crossing. For example, if you take, let's say you take Riesling, you take Madeleine Royale, you cross it, you obtain Müller-Thurgau. This is another story. Hybrids are obtained with the same technique, but you always have Vitis vinifera crossed with another Vitis species, or even, I should add, two other Vitis species crossed together. Most of the time we have a European variety crossed with, let's say, an American Vitis variety, Vitis labrusca, Vitis berlandieri, and so on, they are not Vitis vinifera, they are not European grapes. Most of the time these hybrids are created because these other Vitis species have resistance against fungal diseases or other stuff that we don't want to, and we try to integrate their resistance in the progeny. But the technique is the same. The technique is described here. I took it from Cornell University in the US, which is highly specialized in grape breeding. The first step is to collect the pollen. So here, either you have a completely male variety. This is grape flowers, and only the male part is functioning. The female part is not functioning. We had a question last week. This is the case of several American species which have female-only plant or male-only plants. It is extremely rare in European grapes. We know 2 or 3 examples of cultivated grape varieties in Europe that are only male or only female. It's extremely rare. Most of the time, we have both the female part and the male part in the same flower. So here the cluster begins to flower. So what we do, we collect the pollen. You can either shake or take with the, sorry, you shake it here, and you take the pollen in a tube here as well, or you take with a paintbrush, a small paintbrush, you collect the pollen in a tube. Then the step 2 is called preparing the female. I'm not inventing it. That's how they call it. I find it a little bit funny. But you need, here you have grape flower. You see all these buds here are the ones I showed you on the first slide. And all these buds have the cap. The cap is the petals stuck together. You need to carefully remove it with Brussels here, with the tweezers. With tweezers, you remove carefully this cap, and you must not destroy the male part. This is called emasculation. So you want to take out the pollen. So you take out the cap and then you cut all the pollen bags. You see, all these white branches have no pollen bags on top. It has all been cut. So now the female is ready. Here you have only the female part that is functional. The male part producing the pollen has been removed. It's a very long work. It takes a lot of time, and each of these flowers can now be pollinated with the pollen of your choice. And each of these flowers will give birth to a different variety. That is very important to understand. Now, pollination, you can either use an entire cluster if you have it and you shake it on top of the female, or you have in a tube the pollen, you have a paintbrush, and you brush the female part. So this is how every grape variety that is obtained by breeding by human beings has been made. It's a long work. Usually they do it for thousands of flowers, and they do it for many years. They obtain thousands of grape pips. Then you must plant all of them and see what's happening. Then is the process of selecting the best. This is how grape sex is working either in nature, in vineyards, or in experimental crossings. Now we go to Grüner Veltliner. Its history and origin. Of course, and luckily, its origin is Austria. It was first mentioned in the 18th century in Austria under the old name Weißgipfler. The name Grüner Veltliner appeared later in the 19th century, the end of 19th century. The name, obviously, it means the green from Valtellina. We have no idea why it was named that way. It has nothing to do with Valtellina, as you will see. Maybe it's because the morphology is similar to Roter Veltliner. I will speak about that later. But the link with Valtellina is not yet understood. It's mostly distributed in the eastern part of Austria here, and the surface in Austria was, the total surface in the world was 18,842 hectares in 2010. It is known under different synonyms, not as many as Pinot you had last week, because it's much younger than Pinot. If the variety is young, it did not have enough time to be propagated everywhere and be named differently everywhere. But in Austria, it has 5 different names at least. Grauer Veltliner, which is color mutation. In fact, it's like Grauburgunder is a color mutation of Blauburgunder. Grauburgunder is Pinot Gris, Blauburgunder is Pinot Noir. Here is the same. Grauer Veltliner is a mutation, but still the same species. It was called Grün Muscatella or Grüner Muscatella, which is confusing because it has no muscat aroma and no link with any muscat. Veltliner Grau, Veltliner Grün, or Weißgipfler, which is the old name. We find it in Czech Republic, Veltlinské zelené, which means Grüner Veltliner translated in Czech. Same in Hungarian, Zöld Veltlini. In Italy, it's called Veltliner, Veltlinské zelené in Slovakia, and in Slovenia as well, Zelení Veltliner. So we see that the diversity of the synonyms is not so big. Here is a map of its distribution all around the world. I got this information. I will show the book cover later. It's a book by Kim Anderson, a professor of viticulture and economy in Australia. And he published this book in 2013. There's a new edition coming out soon called Which Wine Grape Varieties Are Grown Where. I'm mentioning it because it's a huge work that is for free to be downloaded on the internet. If you want to have the PDF, it's for free, and I strongly recommend you. So you look at the world total here, 18,842. There are some traces in the US. It has been planted a few decades ago. We count 8 hectares in South America, um, most likely in Argentina. We have some traces in Australia. You know, in Australia, they have a tendency to try almost everything they can. Austria is, of course, the place where it is the most planted. Slovakia, 2,000 hectares. Czech Republic, 1,500, and same in Hungary. So this is quite interesting if you want to compare the different expressions of the grape and the wines in these various areas of Western Europe. Now we move on to the origin of Grüner Veltliner. We knew that it's not coming from Valtellina. It has never been cultivated in the Italian Valtellina, which is north of Lombardy. It has never been observed there. It's close to Südtirol. It has never been observed there historically as well. We know it does not come from there. What we knew since a few decades, since we started to analyze the grape DNA, was a relationship between Savagnin and Grüner Veltliner. We had the hypothesis that Grüner Veltliner was a progeny of Savagnin, but the other parent was missing. So there was a question mark. And when you have it, you can always reverse the relationship. You can tell Savagnin is the parent or Grüner Veltliner is the parent, as long as you don't have this question mark here. But Savagnin is much, much older than Grüner Veltliner. It would make sense to have it as a parent. So that's what we had in mind until 2009. In between, in 2000, an isolated, unknown old vine has been found in St. Georgen am Leitergebirge. This is here, close to, so Vienna is up here, Eisenstadt, Neusiedlersee. It's very close to Eisenstadt, a small village called St. Georgen, which is here. And on this slope here, in abandoned vineyards, they have found an isolated vine. This man here, Mr. Michael Lebel, in 2000 found this vine. It goes much further in the picture you'll see later. Found this vine that was isolated, unknown, and probably a remnant of a former cultivation in this area, maybe more than 100 or 200 years old. This vine was analyzed by a research center. It was identified, and then in 2009, my colleague, Dr. Ferdinand Regner, working at Klosterneuburg, he first identified the vine as not corresponding to any other known variety. And then he started to make DNA parentage analysis, which is much longer and cost-effective than simply identifying. And in 2009, he was able to publish this parentage saying that Grüner Veltliner, here I have pictures with all the grapes and leaves. It's interesting to see the similarities or differences. Grüner Veltliner is a natural crossing, a natural progeny between Savagnin, Traminer, we knew that already, and this obscure grape from St. Georgen. They called it St. Georgener Rebe because nobody knew, and still today nobody knows, which official name it should have. Nobody knows how it was called in the past. But this grape, you look at the leaves, we don't see very much in the picture, but you compare with Grüner Veltliner and Savagnin, it's a mixture. Grüner is a mixture between the two. And this was a fantastic discovery because the missing link in the Grüner Veltliner parentage had been found. And this is thanks to Dr. Ferdinand Regner and also to Michael Lebel, who observed the vine first. So what do we have? We know it does not come from Veltliner. We know it's a natural cross between Savagnin and St. Georgener. We know that it is the child of a single vine in St. Georgen, which is a bit surprising to me, because if we look at this St. Georgener, if it is the parent of Grüner Veltliner, how is that possible that only one plant has survived? This is a mystery to me. And this here we see Mr. Label showing the place. I know that they have run across all this area and everywhere to look for other plants of this St. Georgener, and they didn't find any. So only one plant left living in the world of the parent of Grüner Veltliner, which I find fascinating and almost incredible to be so lucky to have found it. This shows that the ancient vineyards and the ancient varieties must be preserved, must be conserved, because we can have missing links and we can have interesting gene resources from these ancient varieties. So here you see the people, the vine is growing up behind the people in the trees. So it was almost back to wildlife. It used to be cultivated, but it's back to wildlife. And you see all the group of people. Here we see Dr. Ferdinand Regner, Hans Moser with his father, who are the people who are making wine out of it. I'll show you later. So this was a fantastic discovery. And many times when you have a fantastic discovery, you have some people that are jealous. And in 2009 and 2011, this vine was vandalized, which I find completely shocking. Some nasty people came with scissors and they attacked the vine and they tried to destroy it. The first time in 2009 was not so hard, but the second time the plant almost died. It was even reported on television. I think ORF in Austria reported that, well, 500 years old, I don't believe it's so old, but let's say at least 100 years old vine, that is the mother or the parent of Grüner Veltliner, was attacked, vandalized, and almost destroyed. I find it properly shocking. Luckily, in 2012, they had already started to multiply the plants. So they had taken cuttings and grafted onto rootstock to plant a new vineyard here. So the variety was saved. I was lucky in 2012 to visit this area together with Hans Moser, who produces the wine out of it. It's not that I had a big belly. This is, it was raining and it was extremely windy. Maybe today I have this belly, but back then, no. So the plant is behind here, climbing up the trees, and it was still alive. It was not destroyed. The new vineyard that had been planted gave the first vintage, the first harvest in 2015. This is the grapes of this variety that is cultivated. And they even created a Patenschaft, which is a sponsorship to help surviving and help reviving this St. Georgener Rebe. I'm very proud to announce that I'm one of the only 2 foreign members of this Patenschaft, and I have 1 bottle of this wine at home, still unopened. I'm waiting for a good opportunity to taste it. So if anyone is coming to Switzerland, maybe we can open one. So this St. Georgener has been cultivated since 2012, the first wine in 2015. Here we see Hans Moser, who is responsible for the winemaking. This is the wine label. And they even wrote a book about the history of this discovery. Here you have Hans Moser and father from Weingut Moser. I think she's the president of Eisenstadt, Ferdinand Regner, and this this person, I forgot the name, helped to write the book. So all these people, all this association is helping reviving this grape variety, which I find absolutely fantastic. This is the book Von der Wurzel ins Jetzt. There are a lot of information about the history of the area, the discovery, the grape itself, etc. So great work that has been done. Now I want to expand the family tree of Grüner Veltliner to give you a broader picture of what we know today. We start on the right-hand side. In green, you have what is from Austria. In blue, what is from France. And we know now that St. Georgener crossed with Savagnin, Traminer, to give birth to Grüner Veltliner. Okay, that's what we have. But Savagnin, just like Pinot, just like Gouais Blanc we've seen last week, is very important. It's one of the founder varieties that has given birth to many different varieties in Western Europe. Savagnin was also determined as the father of Silvaner, crossing with Österreichisch Weiß, a very old Austrian variety. Silvaner is, of course, very important also in Germany. This one crossed with Roter Veltliner to give Frühroter Veltliner, and Roter Veltliner itself crossed with Savagnin to give Rotgipfler. So what do we see here? Rotgipfler is the son of Savagnin, and Grüner Veltliner is also a son of Savagnin. So these two have one parent in common, so they are half siblings. That's why maybe it used to be called Weißgipfler. Grüner Veltliner used to be called Weißgipfler. An explanation. Now, what is the future of Grüner Veltliner? So I took an example of a bottle of my friend Willy Bründelmeier. He was with us last week. I think he's with us today. So we say hello to Willy, who is one of the famous and excellent winemakers in Austria doing Grüner Veltliner. And sorry, Willy, I made a fake label for your wine because it will be your Ried Käferberg Erste Lage 2120. Okay, I'm not good at label faking. It's just to ask the question, which Grüner Veltliner will we cultivate in 100 years from now? As you know, Grüner Veltliner, like any other variety, is propagated by cuttings or layering. We cannot go through sexuality because if we cross Grüner Veltliner with something else, we obtain a different variety. With climate change, I will address this topic maybe in a future webinar. With climate change, we have an increase of the fungal disease pressure. So powdery mildew, dowdy mildew would become more and more a problem. Here is a map in red of the regions where it will be problematical to cultivate grapevine by 2050. Maybe it's not as a catastrophe as it appears, but it shows that things are changing. So what do? We keep Grüner Veltliner or we take another variety. Here you can, this is we can say the ripening season that is best for every grape. Müller-Thurgau here, you have Nebbiolo here. We can replace with late ripening varieties. We can replace with crossings, with hybrids, as I've shown before. This is a possibility. But if you want to keep Grüner Veltliner, what do you do? You keep doing like this. Of course, on purpose, I took this picture. This is not in Austria, don't worry. But we could continue with agrochemical weaponry. So to put a lot of fungicides, a lot of chemicals to fight against disease, or we could screen the existing clones. That means in the diversity of the grape, we can find some types, some clones that are resistant. It's a long work. Or we could modify Grüner Veltliner genetically, so GM Grüner Veltliner, which is a very sensitive question. I will address it later. So if we want to take another variety here, we can make crossings. It has been done already in 1929 and 79. And my colleague Ferdinand Regner has used these ancient crossings to select some that are resistant to powdery and downy mildew. You might know that Grüner Veltliner is very sensitive to these fungal diseases, and the idea was to obtain the resistance while keeping the character of Grüner Veltliner. So they crossed with, these are hybrids, Malvarina is a hybrid, Cheval Blanc is also a hybrid. They made dozens and dozens of crossings. With this one, no good result, but with this one, they obtained Donau Veltliner, which has been released in 2016. I do not know if it is commercially used by now. I've never seen it on a bottle, but it is authorized to use it. It's a good thing because with that kind of resistant varieties, you can lower very much the use of chemicals in the vineyard. You can much easier have biological agriculture, organic agriculture. It's much better for the environment. So it's a good thing, but is people ready to switch to this? Well, we can ask ourselves the question. This is the book by Kim Anderson I showed you, and I took from this book with all the statistics in the world, I took the sum of all the hybrids and crossings that exist in the world. So the first crossings were made in the late 18th, early 19th century. If you take, for example, Müller-Thurgau was obtained in 1882, which is one of the most successful. Hybrids came a bit later, by the end of the 19th century. And all these hybrids and crossings that have been around since 100 or 200 years, today they cover only 4.4% of the world surface. It's good, but I cannot call it a revolution. In my opinion, people will want to stick to what they have for a long time. I'm sure that Willy and his great-great-grandchildren will want to stick to Grüner Veltliner. I'm sure that Aubert de Villiers and his descendants and family at Romanée-Conti will want to stick with Pinot Noir. So if we want to stick to it, there are a few solutions. One of them is GMOs. I'm not advocating in favor or against. I just want to show you what has been done yet. There are some other possibilities. I will address them in a future webinar when we will talk about climate change. But it's too big a topic to go deep today. I just want to address the question of GMOs, so genetically modified organism. First of all, the possibility to have GMOs is also linked to the fact that we now have the complete genome of Pinot, which means we have read the complete DNA content of Pinot from the beginning to the end. It was done in 2007 by two different teams, one in Italy, one in France. The total cost was 23 million euros. And I find it absolutely fascinating and impressive to know that today it would cost 500 euros. I find it unbelievable. So Pinot, so the grape, was the fourth sequenced plant in the world. We have identified 30,000 genes total. 14,000 have known effect, like powdery mildew resistance, for example, and we can improve selection and physiological monitorage. It has a lot of implications. Also, GMO, so genetically modified organism, is any organism whose genetic material has been altered using genetic engineering techniques. So it's a broad definition. It involves manipulating the DNA molecules. It involves also taking the DNA from one species into the DNA of another one. And the new DNA is called recombinant. It can be, I don't want to go into much details, it can be cisgenic or transgenic. Cisgenic, it means you remain in the same species. You take one gene of grape, you put in another grape. This is cisgenic. Transgenic, it means you go across the species. And if you want, you can take a gene from an elephant and put it in a grape. It is working the same. So these are the two main versions of it. So again, I'm not in favor or against. It can be used very badly, like it was done by Monsanto with Roundup Ready plants. That means they modified corn, soybean, cotton, etc., so that they are resistant to herbicide, and they were selling the herbicide. So they sell the plant seeds and the herbicide of it. So they take agriculture into hostage, and we know the result. There are a lot of documentaries about all the bad effects of it. But it can also have good effects. Here was in Time magazine. This is a Swiss researcher from Zurich University. They created golden rice, which contains vitamin A. In fact, it's a precursor of vitamin A and a lot of iron. And it was used to save the lives of millions of kids, especially in Asia, where they are lacking vitamin A in their food. So it had good intention. It was released for free, so there was no intention of making a lot of money. Yet it was not approved by everyone. Greenpeace, for example, was against this solution, saying that instead of giving them what they are missing, we should improve their own level of life and their own agriculture. Anyway, it's always controversial when we attack that kind of topic. This is also important to understand the labeling and bans. In the United States and Canada, it is not required to label genetically engineered food. So when you travel to US or you live there, you have no possibility to be informed whether it contains GMOs or not. And it's the case since many, many years. Whereas in the green countries, yes, it is significant to be labeled, and there are a lot of bans against GMOs. So you see that worldwide is not so easy. In vitis, some studies have already been done. It could solve mildew, leaf roll virus, Spear's disease, drought, cold resistance, etc. Here's a list. I don't go into detail, but you see in Romania, Germany, USA, China, etc., they tried to make virus resistance, fungal resistance, etc. Many of them have been interrupted because many people were against it. In France, they were working on rootstocks that would be resistant to leaf roll virus. Only rootstocks, not grapevines, not Pinot, Chardonnay, whatever, only rootstocks. And in 2005, they started the project. 2009, it was destroyed by a group of people. I think it was destroyed twice in a row, and the researchers chose to abandon the project under the people's pressure. Here you see angry people saying no to GMO, I don't put GMO in my wine, etc. So it's quite emotional. It's an emotional issue. But as a scientist, we must not get too emotional. We must analyze the data. So these people who angrily fight against GMO, I hope that they do not have in their family someone with diabetes, because 100% of the insulin that is distributed to diabetic people today is made from GMO bacteria that go through fermentation and produce insulin. So this is one example of good use of GMO. I think for me, it's a mistake to say everything black, everything white, everything good, everything bad. We must always think case by case. For example, here, I always tell people, if you eat meat, especially from Argentina or Brazil, or if you drink milk or dairy products, you have a high probability to favor GMOs, because 93% of soybean that is grown in the US and South America, 93% is GMO. And this soybean is exported to Europe and other countries, where it is given as food to the cattle, to the cows, to the pigs, and so on. So the meat that you eat initially is based on GMO food. So if you want to be completely against, you need to be consistent and you need to stop drinking or eating everything that has a tiny link with GMO. And you see that it's not easy because the conversion of all of it takes meat product, egg product, dairy, etc., etc. Again, not in favor, not against, just tell people, inform yourself and be consistent with what you do. So I will finish with a few slides very quickly about gene editing. Gene editing is a technique that is not really considered GMO, although it is quite debatable. They were able to not put it under the GMO umbrella, but still it is manipulating the DNA molecules. The scientific name is a bit weird, clustered regularly interspaced short palindromic repeats. I'm not going into details. It's derived from natural process found in bacteria, and they use it to protect themselves against pathogens. It targets genes for editing and regulating. It's comparable to Photoshop. It's like if you have a gene, you can activate on or off. It's like to switch on or off the light, more or less. It's more complicated, of course, but it's like photoshopping the DNA to make it the way we want. We are not introducing anything new. We just switch on or off. It's a new technique. It's very recent. The first studies in grapes, there are not so many, started in 2016 only. So I don't want to go into many details. It's a bit complicated, but they took Brachetto grape and they tried to knock down, so to mute the mildew locus, and they were able to do it, but only in protoplast. Protoplast are small cells in culture, in laboratory, that are isolated, and they were not able to grow it into a plant. So only protoplast. They had to improve it. So same year, they were able to knock out a gene in Chardonnay, which controls the synthesis of tartaric acid. But again, they were not able to regenerate a plant. So it was only in lab, only in small tubes, and they were not able in 2016 to obtain a plant with this technique. Another, 1 year later, about Neomuscat, was able to change a gene that is responsible for the chlorophyll fabrication, so the green part of the plant. And you see that some plants were albino, so white, and they did not have the chlorophyll. So they were able to do that. And the last study, it looks like it's complicated, but they were able in Thompson seedless, which is Sultanina, a table grape, to make it resistant to Botrytis cinerea, which is the gray mold. So here you see on top the wild type, not resistant, then a few resistant, and the last lines almost completely resistant. And they were able to regenerate the plant. So 2 years ago only, they were able to have plants that were treated with this gene editing technique that looks very promising because it could solve many problems. I know that there are a lot of current studies on grape berry development, sugar content, tartaric acid, aromas, biotic resistance, tolerance for cold and frost, drought tolerance, etc., etc. Fungal disease, of course, we have seen already. So it's a very promising technique. As long as it is still allowed to do so, people will continue to search, and that would be a possibility one day maybe to have a Grüner Veltliner that is finally resistant to fungal diseases. Before I finish, I want to say that all these techniques are very exciting. I don't see any problem with human health, especially with gene editing. The problem that I see is that that kind of techniques are very expensive. This will be done on 1 or 2 clones of Grüner Veltliner or Pinot or Chardonnay. And if it's working, people will jump on these clones and all the other clones, so all the other biodiversity of the grape variety is at risk to be lost. So that's my fear, because I'm always defending the biodiversity and the heritage that we have since generations. So I finish here. I thank you for your attention, and I will close my screen sharing.
Perfect. Thank you, José, for this presentation. And as always, you're quite good in time. And Alexander Stark just wrote a comment to me that this year's Nobel Prize award to Emanuele Carpentier and Jennifer Doudna was in connection of this discovery of the CRISPR-Cas9 you mentioned before. And this was partly, the work were partly done in Vienna.
Yes, because this technique is very promising, especially in human health, in human medicine, because it could solve many diseases that have a genetic basis. So it's very important.
And we already have one question from Roland Graf. Roland, can you unmute your mic, please, and ask your question?
I hope it works now.
Now we can hear you, yes.
Okay, great. Thanks, Bernd. Now, thanks for the seminar so far, but there was one interesting point when you mentioned the first crossing of making Veltliner resistant in 1929 with the Malvania grape. So to my understanding, the first resistant grapes were developed in Russia and in Germany almost during the Second World War. So this is a very, very early date that you mentioned with the Malvania. Have you got any information how it was coming that it was that early that they were trying to make Veltliner resistant?
Well, first of all, thank you for your question, but let me be more specific in what you say. You say making Grüner Veltliner resistant. It's not exactly what they are doing, because when you cross Grüner Veltliner with something else, the result is not Grüner Veltliner anymore. So they were trying to obtain varieties with one of the parents being Grüner Veltliner that would be different, but with the same taste and qualities as Grüner Veltliner, plus the resistance. And this has been done since even earlier than 1929. I know that there were two different breeding programs, one in 1929, one in 1979. I just wanted to show you this. I did not make any research to know all the other programs of breeding using Grüner Veltliner. These are only two examples. It doesn't mean that there are no other crosses earlier or later.
Yeah, there were some done in Switzerland earlier. You're right about that. But do you know where that took place with the Malvania? Because it was the first time that I heard about it.
I could find it. Wait, it was. I think it was done in Klosterneuburg.
Ah, okay. So it was an Austrian research. Yeah. Thank you.
Yeah, you're welcome.
I think there's another comment. Clara, do you want to mention what you have written to me on the Malvarina grape?
Sure. Can you hear me?
Yes, I can hear you, Clara.
Okay. So maybe that's a mistake, because Malvarina was actually crossed in Czech Republic only in 2001. So there was maybe a mistake somewhere. Malvarina is a Czech crossing, and I just checked it, because when the question was posed, I was actually thinking about it, and it was crossed between Villard Blanc and Grüner Veltliner and Merlan in 2001. So it was done in VinSelect. So probably there is a confusion somewhere.
Okay, thank you for the information. I will double check. I took this information from a paper by Ferdinand Regner about these crossings. I will double check.
Yeah, because it didn't exist in 1929.
Yeah, I will double check the date.
Sure, no worries.
Thank you.
You're welcome.
Is there anyone else who would like to raise a question? So perfect. José, thank you for your presentation. All the others for your participation. Later this afternoon, today or tomorrow morning, we will post the slides and the replay function on our website, maywines.com, under the bracket service on the right-hand field. You can find it. Thank you for your participation. All the best and have a good Christmas time. Maybe we see each other next year when we continue with this. Bye-bye.
Thank you.